Welcome to our Microbiology Journal Club. This is the first journal club of 2018. Very exciting. And so today we are going to be talking about this paper, which you can see I've made notes on, which is contrasting patterns of genome level diversity across distinct co-occurring bacterial populations. So I thought this would be really interesting in terms of thinking about microbial ecology. And it's outside of my wheelhouse, so I had to work a little bit to kind of understand what was going on. I read it twice, so I could be sure that I knew what was up. So we'll get launched into this, but before we do, we'll just introduce ourselves. My name is Laura Williams, and I'm an assistant professor at Providence College in Providence, Rhode Island. And then next to me in the little group of windows is Nicole. So Nicole, do you want to introduce yourself?
Yes, hello everyone. I'm Nicole Sectio, and I am a sessional instructor at the University of Northern British Columbia in Prince George, British Columbia, Canada, where I have been tasked this semester with helping undergraduates appreciate microbial life. And that is my life this semester.
It is a noble and worthy cause.
I agree.
All right, and then next to Nicole, we have Elena. Elena, do you want to introduce yourself?
Sure. My name is Elena Zykova, and I'm a bioinformatics scientist at Metagenome Bio in Waterloo, Canada. And I work with metagenomic data, microbiome data, and delving into the world of machine learning right now. So that's exciting.
Excellent. So you're going to be able to help us when we get to all the metagenome stuff in here, I think. But
I'll try to.
Excellent. Okay. So I thought I would start by a place that basically ended up confusing me. And so it's like a broader conceptual idea. So this paper kind of starts out by talking about, okay, we know that bacteria are important members of ecosystems. We know they have effects on biogeochemical cycles. We know that they're an important part of biomass, but we don't know a lot about how bacterial populations within these ecosystems are structured. And I thought, okay, I'm on board. And then they talk a little bit about, you know, we don't have good ways to define ecological populations. And I think this is the part that I wanted to take a minute and kind of talk about where you guys are in terms of your understanding, because they'll come back to this. They keep using these two ideas, like you have genetically distinct populations and then ecologically distinct populations. And my general impression from the paper and the way it was written is that nobody really agrees on how to define ecological populations. And you could even get lots of really healthy fights about how to define genetically distinct populations. But I wanted to see, like, from where you guys stand and what you're working on, does that seem like an accurate read? Or what are your thoughts about this idea that they're trying to nail down with these ecologically distinct populations?
I'm going to come at it from the why can't we all get along type angle. because you have no choice if you're going to have a conversation about this business. So where the paper does end up going is they have chosen to frame ecological distinctness on the basis of genome similarity by sampling time, which is a nice, large, convenient sort of framework, because if you're seeing fluctuations, you can always go back, if this research can continue, and figure out what the environmental correlate is. So ecology starts with thinking about the environment-organism interaction. You're playing on hard mode if you're dealing with metagenomes, because the boundaries of what organismal is, depending on where you want to go with names, is a little hairy for reasons I'm not going to get into, because I'm not very good at talking about that. But there are very many things that you can go and pick apart at to kind of look at the parameters that might be poking your population to stratify a certain way, or making things hang out in high abundance and drop out at low abundance later. So they've chosen what they've chosen, and I think they do a really good job in this paper of sort of trying to have operational definitions that they attempt to use consistently for genome similarity versus the changes in populations by their tribes and their SDP type terminology. So I think this is one of those cases again where like you're naming the things in the context of your study, but you know that somebody might be ready to fight you on your category later, but it's okay. That's sort of where I'm at with that.
Yeah, and just to add on to that, I think it is kind of there is really no definition, and I think because it is different for different species too. Like, what level is going to be ecologically significant for X species is not going to be the same as a Y species. So I think these operational definitions are something that I think generally the community seems to agree on. Is not the numbers for the operational, but you're like, as long as you can define it, and then somebody can look at how you defined it, they can go and define it somehow else, or then try your definition and see how that matches up. But I think there's definitely not a consensus for what the numbers should be, but I think there is a consensus to just define it as you see fit, because it's not going to be the same for different species.
Yeah, I think both of you made really good points on that. And I think that makes sense that, because it's like you said, you're going to have different people defining different criteria so that they can call this as an ecological population that's distinct from this. And that's an excellent point, that if the data is out there, which I think they did release all their data, if you have your own favorite cutoff, then you can go into their data and go, okay, well, if I use my cutoff, then what do I see? And how does that change any possible conclusions? And I think it's interesting because I can see how the boundaries of these ecological populations can get really fuzzy. And we'll get into it in the data, but it's one of the things they did to try to see, are things ecologically distinct, is they said, okay, well, we've got these groups that we've defined by these genetic similarity cutoffs. So what we're going to do now is take time series data and figure out, do some of them like not correlate with each other over time? And that way we can go, aha, they're ecologically distinct. But then they had a data set that pretty much just correlated, and then they got to go, I don't know, not those, at least for now. So I think that's an interesting, it actually, because I don't think about this for my own research, and I don't have training in microbial ecology, the first time I read this, I really struggled with the introduction because I felt like the concepts weren't well defined. And I think it's just because the concepts are not well defined. And I think they're providing some of the definitions as they go. And so for me, reading it, I was kind of like, well, I don't even know what we're talking about right now. And that's because I think this is an open question for a lot of people who are working on this. And they kind of have to decide, here's what we're going to say, here's how we're going to make these cutoffs. This is the operating, principles for this study. Any other thoughts on that? Okay. All right, so here's what they did. They've got 4 freshwater lakes that they have been working with. And so they are looking at microbial communities, or bacterial communities specifically, in these 4 freshwater lakes. And what they're going to do is focus on 2 specific lineages. One is in the Actinobacteria, which they call AC1, this lineage AC1. And then one is in the Alphaproteobacteria, it's ALF, I guess, ALF5. But within that, they're basically looking at a tribe that they're going to call LD12. And so here, I made a note for myself so that I could remember how they're doing these cutoffs. For the purposes of this paper, and I don't know how much, if this is a widely used definition, but for the purpose of this paper, they're calling a tribe a group that the 16S ribosomal RNA genes are greater than 97.9% identical. So we're looking at high 16S ribosomal RNA gene sequence identity for a tribe. So what they've got is they've basically got a whole bunch of single-cell genomes. So I thought this was cool. I've seen some single-cell genomics pop up in other papers, but I haven't seen a lot of it. And I think a lot more people are probably going to push that way, because it's a great way of sampling an ecosystem where you feel like maybe you're not successfully culturing a lot of the members. So then what you can do, and I looked at it, they've used like a fluorescent cell sorting method to actually get individual cells, and then they use amplification to get as much of the genome sequenced from that single cell as they possibly can. And so they came up with this big old table where they've got 33 of these single cell genomes, and they represent 15 tribes, most of which I think are in the Actinobacteria, and then some in that Alphaproteobacteria, and then some that are off from somewhere else. So what they did was they then looked at, they started out by looking at just a phylogeny, which is figure 1. So figure 1A and figure 1B, they just did a single copy genes, and they looked at a phylogenetic tree of representatives of some of these tribes just to get a sense of how do we think they're all related to each other. And it is at this point that they introduced this idea of sequence-discrete populations. And so they're looking at, and I'll pop over to the paper, maybe. There we go. Okay, so they've got, all right. Can you guys see that? Okay, so up here on figure 1A and figure 1B, they're just showing us a phylogeny with some of the representatives of these different lineages within the tribes. And they've got over here, it's L06 and A23, so down in the bottom part of 1A. And they've got a little vertical line, and they've got, ooh, that's a sequence-discrete population. And so they've defined that as being something that has global average nucleotide identity of greater than 95%. And you can see from the tree on there, this is what kind of puzzled me. I was like, oh, that's interesting, because the P03 over there is actually more closely related in this phylogeny, but they've left it out of the sequence-discrete population. So it must be that it's got more regional variation across the genome than the other two, and they didn't put it in there. One of the things that jumped out at me from this is that you've got a lot, the symbols are different lakes. And so you've actually got, even though these lakes are separate from each other, you've actually got these tribes you've got representatives that look like they're pretty closely related to each other and occurring in these different lakes. I looked back at this as I was going through, but I think those were the main things I got from this part of the figure. Did you guys have any thoughts about these two trees? Or the sequence-discrete population thing?
Yeah, I mean, again, this is stuff that I am not very comfortable with, but I guess my question about the point you made about P03 is it's regional variation within the window that's used in the algorithm that they're calculating average nucleotide identity in, right? So I mean, if your window is set, yeah. So it could also be something that would change depending on whether your window was differently sized, I guess. I'm just wondering that. Is that a thing that happens?
Oh, I don't know. That's an interesting thought. I mean, another point that would be good to make here is that because these are single cell genomes, they're not complete. So they are fragmented. And they do mention that is
something I forget. So thank you for reminding me. Yeah,
they do mention that they do have a way to try to estimate genome completeness. And some of these are as low as 40% complete. So they're working with the data they've got, but they are missing some parts of the genome. Maybe that has an impact too.
Yeah, I'm kind of wondering what Elena's thoughts are, because this paper inspired fear in me in terms of my ability to contribute to discourse on it. So I would love to hear from someone else.
Totally no need to have any fear about that. I was actually wondering about the genome completeness as I was looking at this figure. I think it would have been really helpful if they somehow indicated, maybe by symbol size or something like that, what the completeness was of each of those bins as they were mapping this on, just because that would influence it a lot. And To be honest, I didn't read the method section in too much detail. I don't know if they describe, I'm just looking through it now. I'm not, if they describe how they picked the regions where they calculated the G, A, and I. But you're totally right, Nicole. Depending on the region they picked, the A and I numbers could be quite different. So if they picked a very variable region or just around an island, then of course that's going to change a lot. I'd assume they did not do that. I'd assume they picked something that's pretty well conserved and maybe has a cluster of these genes that are used to estimate completeness, like the single copy or little operons that are that co-occur together that are used to define lineages. So I can't see the detail of how they picked that region here.
Yeah, I didn't catch that either. That's a good point.
Yeah, I'm just looking at the A and I section, and they don't really say what regions they selected, but I mean that would be an important thing to know.
And so I think for myself, because I think I lost track of this when I was reading it the first time, and I had to remind myself that the first part of this figure, the A and B, those are conserved single copy genes. So I think that's gene content that's represented in everybody. And then, although I hadn't mentioned it before, the panel C, which is the one to the right that has all the delightful colors on it, is a plot of the genome-wide average nucleotide identity. So that's the genome-wide average nucleotide identity, I guess, against the 16S ribosomal RNA gene identity. And so one of the questions is when they're getting this genome-wide average nucleotide identity, they're going to be missing the fact that it's a draft. They're going to be missing some parts of when they're calculating that number. If you only have a 40% complete genome, that number is going to be missing 60% of the genome that would potentially play into it at that point. But yeah, I don't know how they did, especially when they get into the sliding window. I don't know how they did that exactly. I will say about figure 1C, this is, I thought to myself, this is actually, maybe not, but I feel like this is a really good illustration for people who are kind of new to thinking about why 16S ribosomal RNA genes do not give you the whole story. So for instance, students in my research lab, we will sequence the 16S ribosomal RNA gene as a way of identifying isolates that we get in the lab, very standard practice. And so sometimes two isolates will come back and they'll have very similar or identical 16S ribosomal RNA genes. And then the students will be like, it's exactly the same thing. No, it might be, but chances are it's not. And I think this is a good figure to show that, because it shows you can have pretty high, what looks like pretty high 16S ribosomal RNA gene identity, but you can still have not, your genome-wide average nucleotide identity is still relatively low on this scale, at least. So if you're looking at the top left of that figure, you've got stuff that's got fairly high 16S identity, but then, there's a decent amount of the genome. You're getting some divergence in the genome. So there's some gene content in there that could be important in terms of functional differences and so forth. The point they made with this one was that if you compare, so this was a little confusing to me, because it's basically a pairwise comparison that they then plot as a point. So it's a single cell genome compared to a single cell genome as a pair. They calculate the genome-wide average nucleotide identity of the pair. They calculate the 16S ribosomal RNA gene identity of the pair, and then they stick a point on the plot to reflect that. And so then they color code all the points. And so all the different tribes, they're showing as having pretty low genome-wide average nucleotide identity, which is, I think, what you would expect. And then I didn't know if it was significant that you get kind of most of the green ones, most of the ones from the lineage B1 seem to have pretty, relatively speaking for this group, kind of low average nucleotide identity. And then you kind of get a lot of little clumps of blue. And they didn't talk a lot about that, but I didn't know if this was a way that you could start kind of delineating things, like drawing circles around stuff that looks like it's kind of clumping together and so forth. Do you guys have thoughts on that?
I feel like, I feel like if you were getting really, really
I like that because you're like, I feel like, That's how
like
I feel.
I don't know if this is a clump ready figure is what I guess I'm getting. Like I feel like this is one of those places where maybe you want your coverage to be close to closing some of these, and maybe you want. My question is about population capture of the tribe, because I feel like I would just be the person who does not want to make conclusions about the correlations. I agree with you about the caveats of your 16S nucleotide identity versus talking about functional stratification in your organisms that you brought up earlier. But I don't know. I don't think I would be the person who would say, yes, we could use this as interpreting stratification, if that's where you were going with that suggestion about the clumping. It's interesting, but like, I really don't know what this represents in terms of total capture of that lineage in each of these lakes, I guess. And I don't know if that was in the paper because I didn't deep read that far, but like that's what I'd want to know first. And maybe that's an impossible question to answer, but I feel like that's something I'd want to have some bit of quantitative information about. That wasn't very articulate, and I apologize for that, but that's how I feel about that question.
Yeah, Elena, go ahead.
Actually, I quite like this figure, and like you, I think it's potentially a great teaching tool, something other than like E. coli that we can point to. That's like, look at all this high, percent identity at the 16S, and I think Nicole made a really valid point, like because we don't know the completion, the completion of all of these SAGs may not be equal, and so are we looking at the same thing. But if you were to use these types of plots and you would account for that by only maybe using regions that are present in both of the things in all the things you're comparing and plotting, then you would at least be even with your sampling of the genome. And I think what is kind of striking is the blues do cluster together a little bit better than the greens. And so you would say like, well, in this particular tribe, there tends to be more variation than in this tribe. And so from this lake or from this lake. And, it just seems like the blue tribes from the different lakes seem to be more similar than the green tribes. And I think there's information in that. And I think there's information in, okay, so these lineages may be evolving faster than these lineages or something along those lines. So I think it's a good overall figure. And I think it's a good figure to maybe start building hypotheses they can then test. But yeah, I quite like this figure, and it is a really good representation of like how 16S doesn't always reflect the whole genome. So yeah.
Okay. Yeah, I think that's good. All right. So we've been talking about in our discussion of this. We've talked a little bit about the fact that they kind of acknowledge. Let me see if I can find. I just had it in front of me. I can find their text here. Ooh, I'm getting all turned around. Aha. So this is their global or genome-wide, I guess. I keep calling it global, but I guess that's the same thing. So their genome-wide average nucleotide identity on this plot. And then I'll stop sharing so I don't make anyone sick when I scroll it. So then what they said is, this is useful, but as people have been discussing, it masks potential differences in sequence similarity of individual genes or genome regions. So they're saying, okay, you could have varying rates of divergence across the genome. That makes sense because if you've had horizontal gene transfer events, there's going to be one particular spot of the genome that's going to look very different, and then the neighborhood around it might not. That's going to get kind of masked if you calculate across the whole genome. So then they did what Nicole and Elena have been mentioning, which is decide to do a sliding window and go across the genome and then basically calculate for this window, what's the average nucleotide identity between, between a pair. For this window, what's the average nucleotide identity between a pair. And then they generate a distribution, like a density distribution, saying of the windows that we did, this number of them had really high average nucleotide identity and this number had lower and so forth. So then they can get a distribution plot. And they talk a little bit about what those distribution plots mean. So I'm going to go to that figure. Let me scroll to it first so I don't cause any seasickness amongst people. And then let me share that. Okay. And so the top figure is the AC1 lineage. And what they did was they took one representative of that and then they did pairwise comparisons against the others. And this one representative, this L06, is the one that they picked out as being in a sequence-discrete population with A23. This is, I think, part of how they're supporting that as well, because A23 is that green, really high peak that is just about as close to 100% identity as you can get. So it's coming from the same lake. And then also, if you were to like look at a whole genome alignment of these two, my guess is that they would look really, really, really similar to each other. There's just not a lot of difference between those two. And so they're making the argument that that's a sequence-discrete population, or an SDP, which they keep using. Then they kind of are like, okay, well, what else do we see? And you've got kind of a spectrum then. So the next most closely related thing to L06 is actually our P03 that we were talking about. So even though in the single copy gene phylogeny, P03 is more closely related to L06, here it's showing that with this sliding window scale thing, it does not look as similar if you're looking across the genome rather than whatever set of like 30 conserved single copy genes they used. And then you kind of got this like low spread out shape of the distribution for everybody else, which I had a hard time kind of concluding what that meant. They kind of said, oh, well, you've got like a peak at 80 or something. I'm like, yeah, but you got like a whole, I was trying to think about on the scale of a genome, what that kind of a distribution, like for the red line, for instance, what that would mean. I mean, it obviously means because there's a really low tail at 100%. So there's not hardly any windows where there's high identity. But then I was thinking, well, have you just had time? So you've just collected a lot of mutations, or do you have some horizontal gene transfer events mixed in there? I wonder what those two genomes actually look like if you were to align them. That's a really detailed question. I don't know if anyone has any thoughts or even thought that hard about this figure. But if you have any thoughts, I'd be interested.
Yeah, I also had that thought when I was looking at that distribution. It's just looking at, let's say, a mummer plot or something, you know, where you've got like all the nucleotides next to each other. And would it be just gaps that are, do you have large deletions of whole regions, or do you then have like insertions? Because or is it just, you know, little silent mutations all over the place? And I don't know. I don't know what this means, but I would love to have seen that alignment. Maybe it's in the supplement, but I don't think it is. But I would have loved to see the alignment for sure. And do they specify how close the lakes were to each other at all? Because it'd be interesting to know the biogeography of this type of distribution.
They did acknowledge that people might be thinking biogeography when they look at this data, and they had a reasoning for why they felt like their data. Well, they just have a statement at the end of the very first section of the results that says the collection was not designed to explore biogeography, but they don't really get into detail about that. I'm not sure. I think two of them were Wisconsin. One of them is in Maine. and one's in Germany. So, but I don't know what the two, they did say, I feel like I did read somewhere that they're not, even the ones in Wisconsin are not continuous with, there's no connection between the two of them. So they are separate, but some are within the same state, some within the US, the others within the US, and then, you know, across the pond, basically. Yeah, that's interesting too. Nicole, did you have any? Nicole's like, please, no, I have no thoughts on this.
No. I actually, some of my thoughts are naive, but there are two things I'm thinking of. And like, it's interesting to think about the biogeography, because the other thing that I guess popped into my head is thinking about Kat Milligan-Meyer's stuff, where it's looking at the gut microbiomes of a bunch of different fish from a bunch of fairly isolated, well, within Alaska, but distinct lakes. So, I mean, thinking about using this approach and retrieving genomes of those microbiota to understand your average nucleotide identity and you know, influence of are the genomes being homogenized because they're in the fish, or is it because of the lake retrieval? Like, this is actually a really cool way to start thinking about putting pieces of data like that together, but also just the idea of like, I mean, there's the more traditional thing that you can do, which is the multi-locus sequence typing. And I mean, if there were a bunch of genes, say, if this was one of those more well-characterized lineages where that's always been one of the ways that they split groups apart, how many of those would you cover? Or what happened if those were all actually really similar, but you still had this distribution? So it gives you all these ideas about the levels of, the healthy levels of skepticism you should have when you decide to look at different discrete regions of a genome or looking at the whole thing. So I think that in terms of like, how do you wrap your brain around this, but also the fact that they're starting with something that's a really good primer for thinking about what happens if you've actually gone and decided that getting metagenomes or getting single amplified genomes are the way that you want to go looking at these communities. Is what do we start with as the descriptor of delineation? And you're going to end up with some stuff like that that's a little bit messy, but again, you can build a hypothesis off of it. So this is really cool because you get to watch this kind of struggle or get these things show up in the data, but at least there's something numerical you can do with it to kind of get you to a place to think about where does refinement have to happen. So it's neat to see both like the whole SDP play out in that plot for 2A and the mess of the red line, or not really a mess, but like something that doesn't fit well with a mind that always wants to stick things in boxes.
Yeah, that's a really good point, because it can, I mean, seeing the shape of this can kind of inform, just like you said, like if you're going to pick selected genes that you're going to analyze, where are they occurring on this distribution? Because if you're picking all of them but on one tail, that might change some of the things that you see. Yeah, you should let Kat know about that. I mean, she may already know about that, but you're right. Yeah, that would be a really cool. It's cool to think about how to apply an approach like this to other systems if you're looking at other communities too. So the lower part of this is basically the other lineage that they're thinking about, this LD12. And they point out that the distribution shapes are quite different from what we saw with the A1 lineage at the top. So there's no obvious sequence-discrete population. The only one that they really point out is the one from the A1 lineage. And then they kind of say, oh, we've got this peak at like 92%. That's kind of not something we saw in the A1 lineage. So one thing this shows you is that we've got one group of bacteria, one taxonomic grouping in the Actinobacteria up at the top. We've got that Alphaproteobacterial taxonomic grouping at the bottom. And you're already seeing like you're not necessarily going to see the same thing. So it depends on your representatives that you're sampling. It depends on the lineage itself. And I thought that was interesting too. So there's no one pattern that really kind of popped out as being obviously occurring all the time with both of them. I thought, at least. So this is the extent of what they do with pairwise comparisons of the single cell genomes. Before I move on to how they used their metagenomic data, Was there anything anybody wanted to mention about the single cell genomes before I pop on to the metagenome stuff? Okay, so then what they're going to do is, and this is a section that starts with diversity of wild populations inferred using SAGs, or single amplified genomes. I thought this was pretty cool. That would not have, I read them at this method and I read their approach and I went, oh yeah, that's a really good idea. That's pretty cool. So what they wanted to do was they wanted to say, okay, we've got 33 of these single cell genomes, but we want to kind of know how well did we sample. So of this, with these 33, how well are we representing the diversity and the variation in the populations in these lakes? So what we've got is a time series of metagenome data. And then what they essentially did for the next figure, which is figure 3, is they picked one sample from their time series, which was the sample that was closest to when they collected for the single cell genomics. Because they figured, okay, we're just going to pick the 2 time points. We did single cell genomics, and then we're going to pick the metagenome time point that's close to that. And then they took single cell genomes, probably, I think they tried to use their most complete ones, because we know that these aren't 100% complete. So I think they tried to pick their most complete single cell genomes. And they basically stacked the reads from their metagenome time point up to those reference genomes. And then they plotted basically what's the percent identity with which that read got recruited to that reference genome. So basically, the way I understood it is you've got your reference genome, and then you can say, we're going to recruit reads that match or have 90% identity. And then you look at the reads that recruit. And then you move your cutoff to like 80%, and then you look at the reads that recruit. And then you move your cutoff to 70%, and then you look at the number of reads that recruit. And then that gives you a plot that looks something like this. Get up there to it. Okay, do you guys see that? Okay, so I thought this was a neat idea. I got stuck on one point that they keep coming back to, which they mentioned in the introduction. And maybe Elena knows something about this. They keep mentioning a coverage gap at 90 to 95% identity. And they call this like a coverage discontinuity that they say other people have noticed as well. And I could not for the life of me figure out why that would be a thing. Is there a reason why we've got that?
So very disappointingly, if there is a reason, I don't know it. I have recently seen it in other papers where they've found things that are fairly similar to each other in sequence-wise, like 96 and above, and then there's like nothing, and then things are similar at the order level again. And so whether it's something to do with the way we're clustering, or I don't know, biologically, I'm struggling for a reason, right? Like you're expecting, even when you think about population diversity, generally we don't think there's going to be a population here and a population here. They're going to be completely distinct. You think there's going to be maybe some sort of like smoother lines, not a lot of gaps between percent identity. So that was surprising to me. Like, so things are similar at the species level and then order, and there's nothing in between. That just seems really, really strange. So it's been in the literature fairly recently, but I just don't know. I don't know a good reason for it biologically. Sorry.
Oh no, actually that helps, because I felt like maybe I was just missing some massive biological reason why you would expect to see that. I'm like, I would have, like you said, I kind of assumed you'd see a spectrum. I didn't expect that you'd get this apparently expected cutoff where you've got really very similar stuff, and then it falls off, and then, like you said, we're on the level of maybe like the orders. And that first plot that we're looking at the top left, I think, is really emblematic of that, where you can kind of see this very clear like bimodal peak that they've got going. And even the one to the right of that, you've got a top peak and then there's like a little dip and then, you know, shifting over to like maybe like, I don't know, that's still in kind of like the 90-92. But yeah, that'll be interesting. I wonder if anyone happens to watch this who knows the reason. Let us know. Nicole, have you run across anything with that?
So I did postdoc in a lab that did metagenomics with Elena, but didn't really come away with the content knowledge that allows me to say anything either impressive or sense-making based on this question. Part of me is sort of wondering about the math of uncertainty once you get to this threshold when you're kind of at high identity. Like, I don't really know why you would have a drop-off like that unless there's just like, if it's based on windows, I guess I can see how you could get a plateau where there's just going to be a bunch of regions of reads that never get there. But like that doesn't really make a whole lot of sense to me. It's interesting that this is something that Elena's been seeing more than once in the literature, because this is definitely literature I never read recently based on the work I've been doing. But I'm very grateful for the explanation that both of you provided on this figure, because I think this was the part of the paper, at least this section, where I was not really quite on top of getting what's going on, and I was a little annoyed that the A through D in the figure caption didn't actually line up with anything in this figure, because I was just like, where C is. But now that I understand what they're doing here with linking the metagenomes, this is actually really cool to kind of see, at least with one sort of non-model reference, what level of variation you're getting, you know, of things that are you know, higher order coarsely related based on genome similarity. So again, this is really, it kind of reminds me a bit of our September Journal Club when we talked about Dave's opinion piece on life identification numbers and like these ideas of approaches to sense making when what you're really dealing with are partial or attempted assemblies of genomes, and like understanding what they mean for potentially biogeographical or just within a sampling area stratification of your populations. So it's neat to see this type of representation graphically of what these comparisons look like when you've got like a SAG to work with.
Yeah, and I think I one of the things that I wish they would have done, because I think what they did is they just is like, for instance, this one at the top left, I think they took all of their I think this is what they did. I think they took all of their SAGs from that one lake, and then they just pulled all their metagenomic reads up against that. So that took me a minute because I was used to thinking about pairwise comparisons. But the other thing that I wish they'd done was they mentioned in the text that they said, okay, well, here's, like, for instance, if we're looking at the top left, we've got this very clear distribution where you've got a peak that happens around, what is that, like 75%, I think they say. So then one of the things that they did was they said, okay, well, we did this with, this was non-competitive recruiting. So what we're going to do now is we're going to say, okay, in this plot that you're looking at, it's possible that one read mapped to or got recruited to multiple SAGs. So it could be that it got recruited to its sequence-discrete population partner, maybe, up here at the really close to 100 peak. But it could have potentially also gotten recruited to one that was from a different lineage, a different population. So they allowed, in this plot, they allowed reads to be recruited to multiple SAGs from this lake. But what they then did was they said, now we're going to do competitive recruiting, and we're only going to let it be recruited to its best match. So if that read recruits to its best match, then we're not going to allow it to recruit anywhere else at any lower identities. So when they did that, I think what they said is, let me make sure I've got this right. So under competitive recruiting conditions, the resulting frequency distributions changed and the fraction of reads recruiting with 60 to 90% identity dropped dramatically. I really wish they would have shown that, because I feel like that's their argument for saying, I mean, what they said was, we wanted to know how well we sampled our population with these SAGs. And the best way to show that is to show the competitive recruitment, where you say, hey, we've still, because what they say in the text is a secondary peak around 80% identity still remained. And it is possible these reads originated from cells belonging to other populations that lacked a representative single cell genome. I felt like that was where they were headed with doing this. And so it was odd to me that they didn't show, like, I would have loved to have had on the left the non-competitive recruiting and on the right the matching competitive recruiting plot, so that you could actually see the change that happens. But I think they buried it in the supplemental material. I'm not really sure why. I felt like that was the most convincing part for their argument about how well they've sampled.
I hadn't thought about it that way, which is really cool. And I also hit a point this week where I was just firmly anti-supplement seeking. But I agree with you that, like, that would have been good to actually have alongside as a comparison. And I wonder, this is not a very scientific comment, but I kind of wonder if part of the reason that it ended up in the supplement is because they're sort of, like, we all know that you're going to retrieve a lot of unknown when you do a study like this, right? I mean, that's definitely just something that's going to come through. From a data handling perspective, I agree that it shouldn't have been in the supplement, because for people who don't really know what assumptions would underlie competitive recruitment, which is everything you just unpacked about just high regions of read identity and you know, lining up with more than one SAG in that pool, you don't want that to skew your interpretation of the level of diversity that's in that thing that you sampled. So yeah, I'm with you on that in terms of including it in part of sort of the exposition that came with this research paper more centrally.
Elena, do you have thoughts on that? Yeah.
Yeah, I completely agree. I thought it should have been in the main and not the supplement. And I'm looking at the supplementary figure of that right now. And the pattern is still there. It's maybe a little bit messier, but I agree. It would have been more compelling to show the competitive recruitment rather than non-competitive recruitment. But I don't know. It could also be, this is not really a science-based comment, but it could also be that that's what the reviewers preferred, and, you know, based on what data was maybe neater. But yeah, I would have at least liked to see both of them. But I think the competitive recruitment makes more sense from a data handling perspective rather than non-competitive. So I was actually surprised that they went with that one in the main paper. I guess they could make the argument that they didn't, you can't know for sure which genome it most likely came from, but I don't know. I think picking the one that has the best hit would be better, unless of course you run into the issue where it has two equally good placements, but then you can just maybe exclude those reads. I don't know. So there's maybe reasons why they went with a non-competitive, and that might be not necessarily science-based as well.
Yeah, I'd be interested, because I thought, for me, I was thinking like that if they feel confident that this technique of mapping metagenome reads to single cell genomes from the sampling site is a good way of figuring out whether or not you've got good representation in your single cell genomes or in your whole genomes. I think there would be a lot of people who would be interested in an approach like that to try to get some sense of how well they've got representation. But I agree with you guys. I'd be curious about why the rationale for why they put it in the supplementary material. Maybe in the end they felt it wasn't as convincing as they wanted it to be, and so they focused on the non-competitive recruitment. This is a lot of data, But basically what they're doing is they're kind of looking at essentially what is the representation of the population. They conclude from this that, for instance, the LD12, the one on the top right, according to the text, they feel like they've got the majority of LD12 cells in the lake belong to sequence-discrete populations that they have represented in their single-cell genomes. So they felt that the LD12 data kind of said, okay, we did a pretty good job sampling with our single-cell genomes. The Mendota data, or the AC1 data, they were like, maybe not as good. We might be missing a couple of things. And then those were the biggest things that I pulled out of this figure, apart from the fact that it's very colorful. So then they get into this idea of ecologically distinct populations. So they've been, up to this point, they've been using sequence similarity cutoffs to define populations and sequence discrete populations. And so now they wanted to ask, do these sequence, and in fact, they write this straight into the text, are sequence discrete populations within a tribe ecologically discrete as well? And so what they're going to do is they're now going to focus on sequence discrete populations. So these really, they're going to define their populations that they're going to track with each other as having a nucleotide identity cutoff of 97.5%. So are highly similar groups. And then what they did was they just, across their metagenome time series, they just tracked them against each other. So they've got sequence discrete populations from these lineages, and they're going to track them against each other in the metagenome data and try to figure out if they see correlation or no correlation. And so in the end, what they conclude is that they've got, so for the top, so the top is an AC1 lineage, and they've got 2 sequence discrete populations that they're tracking over time. And they called this a poorly correlated one. And so they've kind of made a tentative hypothesis that they could be ecologically distinct because they're not tracking in abundance with each other over time. For what reasons those are, we don't know. This is where I started to go, wow, that's super fuzzy. But I can understand what they're going for, but I still found it unsatisfying just because I was hoping for something more definitive. But I fully recognize that in microbial ecologists are probably for right now going, yes, and we do as well. But this is where we are. The bottom panel, those populations, they argued track with each other pretty well. I think you can see that in the patterns. And so this is a case where, at least for temporal variability, they were like, we can't say anything about these. So are they spatially distributed differently? I have no idea. Are they distributed differently because of things like changes in oxygen content? No idea. But the best thing they had with their time series was just, do they vary correlated or not correlated with each other over time? Maybe yes on the top, no on the bottom. What that says about ecologically distinct populations, I don't know. What do you guys think? Elena's making a great face.
I mean, even just looking at the top one, like where the red line peaks, the little green line kind of peaks too, like in halfway through 2010 and, you know, the end of 2009. So I don't know if they're that different. Maybe fold change would have been nice to show too. I guess it would have been really cool to see if there was any strong patterns, but maybe they're not differential distributed. Maybe they are. Maybe they're just, you know, a mixed population, and they occur as a mixed population, and that's just to maintain some sort of genetic variability in mixed populations and like this community of this one species. I don't know, but yeah, I wish there was a more pattern than, like, you know, something that a 3-year-old drew with a crayon. That's kind of what it looks like right now. Yeah, I don't know what they can say about that. I don't see too much correlation with either one. I mean, they could be not responding to each other at all. It could be just conditions, right? So, you know, they like to do, you know, they bloom at the same times, they crash at the same times, and that's kind of I don't know.
Yeah, true. Nicole, did you have any thoughts on this?
I'm with Elena on that. I mean, these are lakes, so I mean, for me, if your water, if your microbes came out of the same piece of filter paper that was dipped into the lake, however you did this, or it came out of the same layer when you stuck your Niskin bottle in, if you go with the whole broad, it's going to be temperature, you know, that sort of thing. You might have growth kinetics that are allowing them to just line up, but not addressing the filter that lets you see the ecological distinctness. So I mean, there's always that big question of like, are we sampling along a variable that's allowing you to see that there are some differences in the number of reads that you see? Like maybe this was just the wrong context. I know that's really broad and sort of a cop-out, but I'm having a hard time with either of these figures. But I mean, I appreciate the approach. You do have to start with something, right? I mean, and having time series data is important because there's also the connectivity of what these populations are doing. So I mean, maybe this is, I'm going to link it to something that's not related, but I mean, what if these fluctuations are just associated with the stochasticity boundary that you have for things not being quite so bad in that lake? So I'm linking it, I guess, to the idea of disease, or if it's sort of like microbial configurations around asymptomatic or well, because there's that continuation before you get to a real community fluctuation when there's a problem. Like, we don't know. And maybe the idea of thinking about distinctness actually has to happen over a longer framework of things that might actually be responses that are not setting a filter apart from them. So, I mean, there's a lot to unpack in that question of how do we feel about the data. I'm like, they did what they could do with it. And I'm glad that it's out there because somebody could see how you might do the synthesis. But yeah, it's hard to tell.
Yeah, I mean, I agree that it was great that they did it, and it's great to see it. But like, I guess, you know, you kind of hope for like these clean ecotypes, and you know, when you don't get that, you're like, oh, I don't know what this means. I don't know how to interpret this. So, but I mean, it's great that they did it. So yeah.
All right, so the last figure we've got, and I know we're reaching an hour, so I'll try to make this quick because I try to don't hold people for longer than an hour. But the last figure they did, which I thought was interesting, is they were looking at each time point for these representative single cell genomes. So they've got a single cell genome, and then they do metagenomic read recruitment, and they track the average nucleotide identity, I think is what they're doing. So they're tracking nucleotide identity of the reads. And I felt like this was edging towards trying to kind of track potential divergence. So if you're starting with reads that are very similar to a single cell genome, and then you start to kind of see a population start to diverge, then you would start to see the average nucleotide identity start to drop. Thought that's where they were going with this. But if you look at it, it's kind of, you know, I mean, it's like, I think this is really interesting. The only thing that jumped out at me at all is actually LO6, interestingly enough, because that was one of the ones that they had as a member of a sequence-discrete population. And that's 1, 2, 3, 4, 5 up from the bottom, because it's the one where I felt like you could see that starts, the metagenomic read recruitment identity starts high, and then as you get to the later samples, it's dropping a little bit. And so there's potentially, and you can kind of see that reflected in the plot to the left, where the percent, you could see it's down a little bit lower, I think. Oh yeah, it starts out kind of high, and then it's got this like tail kind of thing. But I had a tough time with this figure. Nothing just leaped out at me like, here it is. And they don't actually talk that much about it either. But it's an interesting way to go through and analyze their data. Did you guys have any thoughts about this figure? Okay.
I think I feel the same way about it as I do with the one we just discussed before, where it's like, it's getting to the point where you you know, it's fodder for figuring out where you're going to go next in your exploration, but like knowing what it means at this point, can't really go there.
Yeah. So since it's 4, I'll go ahead and have us just do a little bit of a wrap up. I think I picked this paper, so I was happy to read it because it helped me think a little bit about how to deal with this kind of data, and it was good for me as somebody who doesn't have a strong microbial ecology background. It was good for me to be thinking about what are the types of questions people are asking. What is the available data that we could use to try to address things like how do you define populations on a genetic or an ecological level, and then different ways that they handled their data were good for me to kind of think about in terms of the metagenome plus the single cell genomics, using those two together. I think that was really interesting. So did you guys have any kind of closing thoughts about the paper?
I really like the paper. And I think it provides a really good framework for how to deal with this type of data. And I think it provides a good framework for where microbial ecologists could be taking the data that we already have, or maybe already using existing data with new data sets to try and to understand really basic things about evolution in the ecological context. So I thought it was a really good paper. I enjoyed it. Yeah.
I'm completely agreeing with everything that's been said at this point. It was very, very invigorating to actually look at a paper that's sort of attempting to do that kind of temporal sense making with the population, you know, retrieved by a metagenomics approach where you can't, where it's actually delightfully divorced from lowest common ancestry and just trying to put things in boxes, but just be like, look, what are the genomes? How are they relating to each other in something that actually constitutes quantitative relatedness, and how is it changing over time? And I think we need to help people understand that moving that way is not insurmountable if you've been trained a certain way, i.e. a taxonomy-centric way, and that this will help, you know, with understanding your system of microbial life, and you know, as long as you've got like a sampling scheme that's anchoring you to something, you can start to do some type of construction and synthesis. So it was really, really nice to kind of move outside of microbiome reading for a bit and just look at this as a framework for sure. So I appreciate the selection. Thank you.
Of course. So, so with that, we'll kind of wrap it up. I want to thank you both for being here. I always look forward to these discussions, and it's nice to have. So Elena is new. This is her first time here. And we always welcome new people. So if somebody has if you have watched this far, you should definitely come join us, because I'm sure you would have something interesting to contribute to the discussion. We have the website is g+microjc.wordpress.com. That's where I post the updates. We're aiming to kind of keep a regular schedule, so we're going to try for the last Thursday of every month at 3 p.m. Eastern Time. At the moment, I don't have a paper picked, so if anybody has a paper on their to-read list, absolutely feel free to send me some suggestions, and I would be delighted to include that in our upcoming journal clubs. So with that, I'll sign off and thank everybody for coming.